Photograph a historic site, help Wikipedia, and win a prize. Participate in the world's largest photography competition this month. Learn more. Peroxisome. 58 languages. Basic structure of a peroxisome. Distribution of peroxismes, white, in HEK. 293 cells during mitosis. A peroxisome, IPA, PKSOM, 1, is a membrane-bound organelle, a type of microbody, found in the cytoplasm of virtually all eukaryotic cells. 2, 3. Peroxismes are oxidative organelles. Frequently, molecular oxygen serves as a co-substrate, from which hydrogen peroxide, H2O2, is then formed. Peroxismes owe their name to hydrogen peroxide generating and scavenging activities. They perform key roles in lipid metabolism and the reduction of reactive oxygen species. 4. Peroxismes are involved in the catabolism of very long-chain fatty acids, branched-chain fatty acids, bile acid intermediates, in the liver, D-amino acids, and polyamines. Peroxismes also play a role in the biosynthesis of plasmalogens, ether phospholipids, critical for the normal function of mammalian brains and lungs. 5. Peroxismes contain approximately 10% of the total activity of two enzymes, glucose 6 phosphate dehydrogenase and 6-phosphogluconate dehydrogenase, in the pentose phosphate pathway, 6, which is important for energy metabolism. 5. It is vigorously debated whether peroxismes are involved in isoprenoid and cholesterol synthesis in animals. 5. Other peroxisomal functions include the glyoxylate cycle in germinating seeds, glyoxysms, photorespiration in leaves, 7, glycolysis in trypanosomes, glycosomes, and methanol and amine oxidation, and assimilation in some yeasts. 007. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Peroxisome in rat neonatal cardiomyocyte. History, edit. Peroxismes, microbodies, were first described by a Swedish doctoral student, J. Roden, in 1954. 8. They were identified as organelles by Christian de Duve and Pierre Bourdieu in 1966. 9. De Duve and co-workers discovered that peroxismes contain several oxidases involved in the production of hydrogen peroxide, H2O2, as well as catalase involved in the decomposition of H2O2 to oxygen and water. 10. Due to their role in peroxide metabolism, de Duve named them peroxismes, replacing the formerly used morphological term microbodies. Later, it was described that firefly luciferase is targeted to peroxismes in mammalian cells, allowing the discovery of the import targeting signal for peroxismes, and triggering many advances in the peroxisome biogenesis field. 11, 12. Structure, edit. Peroxismes are small, 0.11 m diameter, subcellular compartments, organelles, with a fine granular matrix and surrounded by a single biomembrane which are located in the cytoplasm of a cell. 13, 14. Compartmentalization creates an optimized environment to promote various metabolic reactions within peroxismes required to sustain cellular functions and viability of the organism. The number, size and protein composition of peroxismes are variable and depend on cell type and environmental conditions. For example, in baker's yeast, S. cerevisiae, it has been observed that with good glucose supply, only a few, small peroxismes are present. In contrast, when the yeasts were supplied with long-chain fatty acids as sole carbon source, up to 20 to 25 large peroxismes can be formed. 15. Metabolic functions, edit. A major function of the peroxisme is the breakdown of very long-chain fatty acids through beta oxidation. In animal cells, the long fatty acids are converted to medium-chain fatty acids, which are subsequently shuttled to mitochondria, where they eventually are broken down to carbon dioxide and water. In yeast and plant cells, this process is carried out exclusively in peroxismes. 16, 17. The first reactions in the formation of plasmalogen in animal cells also occur in peroxismes. Plasmalogen is the most abundant phospholipid in myelin. Deficiency of plasmalogens causes profound abnormalities in the myelination of nerve cells, which is one reason why many peroxisomal disorders affect the nervous system. 16. Peroxismes also play a role in the production. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF of bile acids important for the absorption of fats and fat-soluble vitamins, such as vitamins A and K. Skin disorders are features of genetic disorders affecting peroxisme function as a result. 17. The specific metabolic pathways that occur exclusively in mammalian peroxismes are 5. Oxidation of phytanic acid. Oxidation of very long-chain and polyunsaturated fatty acids. Biosynthesis of plasmalogens. Conjugation of cholic acid as part of bile acid synthesis. Peroxismes contain oxidative enzymes, such as D-amino acid oxidase and uric acid oxidase. 18. However, the last enzyme is absent in humans, explaining the disease known as gout, caused by the accumulation of uric acid. Certain enzymes within the peroxisme, by using molecular oxygen, remove hydrogen atoms from specific organic substrates, labeled as R, in an oxidative reaction, producing hydrogen peroxide, H. 2. O. 2. Itself toxic. Catalase, another peroxisomal enzyme, uses this H. 2. O. 2. to oxidize other substrates, including phenols, formic acid, formaldehyde, and alcohol, by means of the peroxidation reaction, thus eliminating the poisonous hydrogen peroxide in the process. This reaction is important in liver and kidney cells, where the peroxismes detoxify various toxic substances that enter the blood. About 25% of the ethanol that humans consume by drinking alcoholic beverages is oxidized to acetaldehyde in this way. 16. In addition, when excess H. 2. O. 2. accumulates in the cell, catalase converts it to H. 2. O through this reaction. In higher plants, peroxismes contain also a complex battery of antioxidative enzymes such as superoxide dismutase, the components of the ascorbate glutathione cycle, and the NADP dehydrogenases of the pentose phosphate pathway. It has been demonstrated that peroxismes generate superoxide, O2, and nitric oxide, No, radicals. 19, 20. There is evidence now that those reactive oxygen species including peroxisomal H2, O2, are also important signaling molecules in plants and animals and contribute to healthy aging and age-related disorders in humans. 21. The peroxisome of plant cells is polarized when fighting fungal penetration. Infection causes a glucosinolate molecule to play an antifungal role to be made and delivered to the outside of the cell through the action of the peroxisomal proteins, PEN2 and PEN3. 22. Peroxisomes in mammals and humans also contribute to antiviral defense. 23. And the combative pathogens. 24. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Peroxisome assembly, edit. Peroxisomes are derived from the smooth endoplasmic reticulum under certain experimental conditions and replicate by membrane growth and division out of pre-existing organelles. 25, 26, 27. Peroxisome matrix proteins are translated in the cytoplasm prior to import. Specific amino acid sequences, points or peroxisomal targeting signal, at the C-terminus, points 1, or N-terminus, points 2, of peroxisomal matrix proteins signals them to be imported into the organelle by a targeting factor. There are currently 36 known proteins involved in peroxisome biogenesis and maintenance, called peroxins, 28, which participate in the process of peroxisome assembly in different organisms. In mammalian cells there are 13 characterized peroxins. In contrast to protein import into the endoplasmic reticulum, or mitochondria, proteins do not need to be unfolded to be imported into the peroxisome lumen. The matrix protein import receptors, the peroxins PEX5 and PEX7, accompany their cargoes, containing a points 1 or a points 2 amino acid sequence, respectively, all the way to the peroxisome where they release the cargo into the peroxisomal matrix and then return to the cytosol, a step named recycling. A special way of peroxisomal protein targeting is called piggy-backing. Proteins that are transported by this unique method do not have a canonical points, but rather bind on a points protein to be transported as a complex. 29. A model describing the import cycle is referred to as the extended shuttle mechanism. 30. There is now evidence that ATP hydrolysis is required for the recycling of receptors to the cytosol. Also, ubiquitination is crucial for the export of PEX5 from the peroxisome to the cytosol. The biogenesis of the peroxisomal membrane and the insertion of peroxisomal membrane proteins, PMPs, requires the peroxins PEX19, PEX3, and PEX16. PEX19 is a PMP receptor and chaperone, which binds the PMPs and routes them to the peroxisomal membrane, where it interacts with PEX3, a peroxisomal integral membrane protein. PMPs are then inserted into the peroxisomal membrane. The degradation of peroxisomes is called pexophagy. 31. Peroxisome interaction and communication, edit. The diverse functions of peroxisomes require dynamic interactions and cooperation with many organelles involved in cellular lipid metabolism such as the endoplasmic reticulum, mitochondria, lipid droplets, and lysosomes. 32. Peroxismes interact with mitochondria in several metabolic pathways, including oxidation of fatty acids and the metabolism of reactive oxygen species. 5. Both organelles are in close contact with the endoplasmic reticulum and share several proteins, including organelle fission factors. 33. Peroxismes also interact with the endoplasmic reticulum and cooperate in the synthesis of ether lipids, plasmalogens, which are important for nerve cells, see above. In filamentous fungi, peroxismes move on microtubules by hitchhiking, a process involving contact with rapidly moving early endosomes. 34. Physical contact between organelles is often mediated by membrane contact. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF sites, where membranes of two organelles are physically tethered to enable rapid transfer of small molecules, enable organelle communication, and are crucial for coordination of cellular functions, and hence human health. 35. Alterations of membrane contacts have been observed in various diseases. Associated medical conditions, edit. Peroxisomal disorders are a class of medical conditions that typically affect the human nervous system as well as many other organ systems. Two common examples are X-linked adrenoleukodystrophy and the peroxisome biogenesis disorders. 36, 37. Genes, edit. PEX genes encode the protein machinery, peroxins, required for proper peroxisome assembly. Peroxisomal membrane proteins are imported through at least two routes, one of which depends on interaction between peroxin 19 and peroxin 3, while the other is required for import of peroxin 3. Either of which may occur without the import of matrix, lumen, enzymes, which possess the peroxisomal targeting signal points 1 or points 2 as previously discussed. 38. Elongation of the peroxisome membrane and the final fission of the organelle are regulated by PEX11P. 39. Genes that encode peroxin proteins include PEX1, PEX2, PXMP3, PEX3, PEX5, PEX6, PEX7, PEX9, 40, 41, PEX10, PEX11O, PEX11B, PEX11G, PEX12, PEX13, PEX14, PEX16, PEX19, PEX26, PEX28, PEX30, and PEX31. Between organisms, PEX numbering and function can differ. Evolutionary origins, edit. The protein content of peroxismes varies across species or organism, but the presence of proteins common to many species has been used to suggest an endosymbiotic origin, that is, peroxismes evolved from bacteria that invaded larger cells as parasites, and very gradually evolved a symbiotic relationship. 42. However, this view has been challenged by recent discoveries. 43. For example, peroxisome-less mutants can restore peroxismes upon introduction of the wild-type gene. Two independent evolutionary analyses of the peroxisomal proteome found homologies between the peroxisomal import machinery and the IRID pathway in the endoplasmic reticulum, 44, 45. Along with a number of metabolic enzymes that were likely recruited from the mitochondria. 45. The peroxisome may have had an actinomystoter origin. 46. However, this is controversial. 47. Other related organelles, edit. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Other organelles of the microbody family related to peroxisms include glioxisms of plants and filamentous fungi, glycosomes of kinetoplastids, 48, and Warrenin bodies of filamentous fungi. See also, edit. Peroxisome proliferator activated receptor. References, edit. 1. Definition of peroxisome. www.merriam-webster.com. Retrieved 30 October 2019. 2. Eilinger M., Voelkler, Fahimi H.D., Schrader M., November 2018. The Peroxisome, an update on Mysteries 2.0. Histochemistry and Cell Biology. 150, 5443471 do, 10.1000/S00418-018-1722. 5. PMC 6182659. PMID 302199925. 3. O'Connell J.D., Chow, Ellington A.D., Marcotte M., 2012. Dynamic reorganization of metabolic enzymes into intracellular bodies. A new Rev Cell Dev Biol. 28, 89111. Do. 10.1146/An of Cell Bio. 101,011 to 155,841. PMC 4089986. PMID 230577741. 4. Bonikampner, Volkler, Fahimi H.D., Schrader M., 2009. Reactive oxygen species and peroxisms. Struggling for balance. Biofactors. 35, 434655 do, 10.1002/biof.48. PMID 194599143. S2 SID 7502822. 5. ABCDE Wonders RJ, Waterham HR, 2006. Biochemistry of Mammalian Peroxisms Revisited. Annual Review of Biochemistry. 75, 295332. Do. 10.101146/biochem.74.0828003.1333229. PMID 167564994. 6. Antonenkov, Vasily D. July 1989. Dehydrogenases of the pentose phosphate pathway in rat liver. Peroxisms. European Journal of Biochemistry. 183, 17582 do, 10.1111/J.1432. 1033.1989.TB14898.X. ISSN 0014-2956. PMID 2753047. 7. Evert RF, Eichhorn SE, 2006. Esau's Plant Anatomy, Meristems, Cells, and Tissues of the Plant Body, Their Structure, Function, and Development. John Wiley and Sons. ISBN 9780471738435. 8. Roden, J. 1954. Correlation of ultrastructural organization and function in normal and experimentally changed proximal tubule cells of the mouse kidney. Doctorate thesis. Karolinska Institute, Stockholm. 9 de Duve, Christian, Bourdieu, Pierre, 1966. Peroxismes, Microbodies and Related Particles. Physiological Reviews. 46, 2323357 10.1152/FISREV.1966.46.2.323. 10 de Duve c. April 1969. The Peroxisome, a new cytoplasmic organelle. Proceedings of the Royal Society of London. Series B, Biological Sciences. 173, 1030-70-183. Bibcode, 1969 RSPSB.173, 71D. Do. 10.1098/RSPB.1969.0039. PMID 4389648. S2 SID 86579094. 11 Keller, Gia. Gould, S. Deluca, M. Subramani, S. May 1987. Firefly luciferase is targeted to peroxismes in mammalian cells. Proceedings of the National Academy of Sciences. 84, 103264. 3268 Bibcode, 1987 NAS, 84.3264K. Do. 10.1073/NAS.84.10.3264. ISSN 0027-8424. PMC 304849. PMID 35542235. 12 Gould, S.J. September 1988. Identification of peroxisomal targeting signals located at the carboxy terminus of 4 peroxisomal proteins. The Journal of Cell Biology. 107, 3897905. Do. 10.1083/JCB.107.3.897. ISSN 0021-9525. PMC 21152268. PMID 29014222. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 13 Carlson, P. Donek D. Kuhlmann J. Fuchs G. Schrick W. 2005. Carlson's Biochemistry and Pathobiochemistry, 15 ed. Stuttgart, Georgtheum. pp. 396f. ISBN 978-3133578158. OCLC 1814744420. 14 Raven PH, Evert RF, Eichhorn SE, 2006. Biology of Plants, 4 ed. Berlin, De Gruyter. pp. 53f. ISBN 978-3-11-018531-7. OCLC 1809043666. 15 Feldman H., 2009. Yeast, Molecular and Cell Biology. Wainheim, Huila VCH. p. 159. ISBN 978. 3527326099 OCLC 4896299727. 16 ABC Alberts B, Johnson O, Lewis J, Raff M, Roberts K, Walter P, 2002. Chapter 12. Peroxismes. Molecular Biology of the Cell, 4th ed. New York, Garland Science. ISBN 978-0-8153-3218-3. 17 A. B. Schrader, Michael, Kamashita, Maki, Eilinger, Marcus, March 2019. Organelle interplay. Peroxisome interactions in health and disease. Journal of Inherited Metabolic Disease. 43, 171. 89 do. 10.1002/Jim.12083. ISSN 1573-2665. PMC 7041636. PMID 30864148. 18 Del Rio LA, Sandalio LM, Palmer JM, Bueno P, Corpus FJ, November 1992. Metabolism of oxygen. Radicals in peroxismes and cellular implications. Free Radical Biology and Medicine. 13, 555780. Do. 10.1016/0891-5849, 92, 90150F. PMID 13343030. 19 Corpus FJ, Barroso JB, Del Rio LA, April 2001. Peroxismes as a source of reactive oxygen species and nitric oxide signal molecules in plant cells. Trends in Plant Science. 6, 414550. Do. 10.1016/1360-1385, 01, 01898-2. PMID 11286918. 20 Corpus FJ, Barroso JB, Carreras Cross M, Leonum, Romero Portas MC, it al. September 2004. Cellular and subcellular localization of endogenous nitric oxide in young and senescent pea plants. Plant Physiology. 136, 2722 do. 10.1104/pp.104.0428112. PMC 523336. PMID 15347796. 21 Lismond C, Ravanco I, Franson M, July 2019. Peroxisomal hydrogen peroxide metabolism and signaling in health and disease. International Journal of Molecular Sciences. 20, 15 3673. Do. 10.3390/IGMS 20153673. PMC 66956606. PMID 31357514. 22 Bednarek P, Pisluska Bednarek M, Svetoso, Schneider B, Daubsky J, Mansurova M, it al. January 2009. A glucosinolate metabolism pathway in living plant cells mediates broad-spectrum antifungal defense. Science. 323, 5910-1016. Bib code 2009 SCI 323101B. Do. 10.1126/science.1163732. PMID 19095900. S2 SID 38423996. 23 Dixit E, Boland S, Chang Y, Liaz, Odendal C, Shum B, it al. May 2010. Peroxismes are signaling platforms for antiviral innate immunity. Cell 141, 466881. Do 10.1016/j.cell.2010.04.018. PMC 3670185. PMID 2045123. 24-DI-F, Bulo MH, Simon Serge, Rashubinsky Ra, November 2018. Dysfunctional peroxismes compromise gut structure and host defense by increased cell death and TOR-dependent autophagy. Molecular biology of the cell. 29, 22 2766 2783 do 10.1016.mbc.e18-07-0434. PMC 6249834. PMID 301886767. 25-Hu D, Schildnet D, Brakeman I, Philipson P, Tabak HF, July 2005. Contribution of the endoplasmic reticulum to peroxisome formation. Cell 122, 185-95 do 10.1016/j.cell.2005.04.025. HDL 1874/9833. PMID 1600935. S2 SID 188370009. 26 Schrader M, Costello JL, Godin Ho LF, Azadias, Eilinger M, May 2016. Proliferation and fission of peroxismes, an update. Biochimica et Biophysica Acta, BBA, Molecular Cell Research. 1863, 5. 97183. Do 10.1016/j.bamca.2015.09.024. HDL 10871/18323. PMID 264094886. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 27 Lazaro Al PB, Fujiki Y, November 1985. Biogenesis of peroxismes. Annual Review of Cell Biology. 1, 489530. Do 10.1011.cb.01.11018.0024221. PMID 3916321. 28 Salim Ra, Smith JJ, Aitchison JD, December 2006. Proteomics of the peroxisme. Biochimica et Biophysica Acta, BBA, Molecular Cell Research. 1763, 12 1541-51. Do 10.1016/j.bamca.2006.09.005. PMC 1858641. PMID 170500007. 29 Toms, Sven, November 2015. Import of proteins into peroxismes, piggybacking to a new home away from home. Open Biology. 5, 11 150148 do 10.1098/asob.1501148. ISSN 2046-2441. PMC 4680570. PMID 2658157 30 Dam IV, Subramani S., April 2001. The human peroxisomal targeting signal receptor, PEX5P, is translocated into the peroxisomal matrix and recycled to the cytosol. Cell 105, 218796. Site CX 10.1.1.385.9484. Do 10.1016/S0092-8674, 01, 00310-5. PMID 11336669. S2 SID 18873642. 31 Abahart T, Kovacs WJ, November 2018. Pexophagy in yeast and mammals, an update on mysteries. Histochemistry and cell biology. 150, 5473488 10.100718-018-018-1724-3. HDL 20.500.11850/302080. PMID 302381155. S2 SID 52307878. 32 Shayin, Stjuldner M., Zilkva E., May 2016. No peroxisome is an island, peroxisome contact. Sites: Biochimica et Biophysica Acta, BBA, Molecular Cell Research. 1863, 1061-9. Do 10.1016/J.Bamker.2015.09.016. PMC 4869879. PMID 2638487. 33 Costello J.L., Passmore J.B., Eilinger M., Schrader M., 2018. Multilocalized proteins, the peroxisome. Mitochondria connection. Proteomics of peroxisomes. Subcellular biochemistry. Volume 89. pp. 383415. Do 10.10174897898113223334_17. ISBN 978-9813-2232-7. PMID 3037833. 34 Salaginis J., Rec Peterson S.L., 2017. Hitchhiking, a non-canonical mode of microtubule-based transport. Trends in Cell Biology. 27, 2141-152, 10.1016/J.TCB.2016.09.005. PMC 5258766. PMID 2766563. 35 Castro I.G., Stjuldner M., Zilkva E., March 2018. Mind the organelle gap, peroxisome contact. Sites in disease. Trends in biochemical sciences. 43, 3199-210. Do 10.1016/J.Types.2018.01.001. PMC 6252078. PMID 293956553. 36 Depreter M., Espel M., Royals F., June 2003. Human peroxisomal disorders. Microscopy. Research and Technique. 61, 220323 Do 10.1002/gem.10330. PMID 127408727. S2 SID 377483992. 37 Eilinger, Marcus, Grill, Sandra, Fahimi, H. Dariush, Schrader, Michael, March 2012. The Peroxisome. An update on mysteries. Histosh Mystery and Cell Biology. 137, 5547574 do. 10.1007/S00418. 012-0941-4. HDL 10871/33969. ISSN 0948-6143. PMC 6182659. PMID 224150027. S2 SID 1485339. 38 Fang Y, Morrell JC, Jones JM, Gould SJ, 2004. PEX3 functions as a PEX19 docking factor in the import of Class I peroxisomal membrane proteins. Journal of Cell Biology. 164, 6863875. Do. 10.1083/JCB.20031131. PMC 2172291. PMID 1500761. 39 Williams C., Opalinski L., Landgraf C., Costello J., Schrader M., Krukkanum, Noops K., Cramham. Volkmer R., Van der Clay I.J., 2015. The membrane remodeling protein PEX11P activates the GT pathway. DNM1P during peroxisomal fission. Proceedings of the National Academy of Sciences of the United States of America. 112, 26,377 6,382 do. 10.1073/NAS.14187361112. PMC 4443378. PMID 259414007. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 40 Felsberg D. Cruz Zaragoza LD, Schlips W., Erdmann R., November 2016. PEX9P is a new yeast peroxisomal import receptor 4.1 containing proteins. Journal of Cell Science. 129, 21 4057. 4066 do. 10.1242/JCS.195271. PMID 276784887. 41 Yafraki, Quartzman SG, Dahan N, Maskit S, Zadar L, Whale U, It al. November 2016. Characterization of proteome dynamics during growth in olete reveals a new peroxisome targeting receptor. Journal of Cell Science. 129, 21 4067 4075 do. 10.1242/JCS.195255. PMC 6275125. PMID 276635510. 42 Lazaro Al PB, Fujiki Y, 1985. Biogenesis of Peroxisomes. Annual Review of Cell Biology. 1, 489. 530 do. 10.100146/anov.cb.01.11018.0024221. PMID 3916321. 43 Figarazen Rashubinsky-Ra, 2007. Maintaining Peroxisome Populations, A Story of Division and Inheritance. Annual Review of Cell and Developmental Biology. 23, 32144. Do. 10.100146/anov.cellbio.23.0905006.123456. PMID 175067002. 44 Schluter, 4 Des, Ripar, Mandel J.L., Poco, Pujola, April 2006. The Evolutionary Origin of Peroxismes, a near-peroxisme connection. Molecular Biology and Evolution. 23, 483845. Do. 10.1093/Molbev/MSJ103. PMID 164521116. 45 AB Gabalden T, Snorby, Van Zimmen F, Hemrica W, Tabak H, Huinen Ma, March 2006. Origin and Evolution of the Peroxisomal Proteome. Biology Direct. 1, 8. Do. 10.101145/6150-1-8. PMC 1472686. PMID 165563314. 46 Duhitar N, Laha, Satoshi S, Kazuo H, Daesu Kem, Takao S, January 2010. The Origin of Peroxismes, the Possibility of an Actinobacterial Symbiosis. Gene 450, 1-1824. Do. 10.1016/j.gene.2009.09.014. PMID 198183887. 47 Gabalden T, Capella Gutierrez S, October 2010. Lack of phylogenetic support for a supposed actinobacterial origin of peroxismes. Gene 465, 1-261-5 do. 10.1016/j.gene.2010.06.004. PMID 206000706. 48 Blattner J, Swinkles B, Dorsum H, Prospero T, Subramani S, Clayton C, December 1992. Glycosome assembly in trypanosomes, variations in the acceptable degeneracy of a Kuhu terminal microbody targeting signal. The Journal of Cell Biology. 119, 1129-36. Do. 10.1083/jcb.119.5.1129. PMC 2289717. PMID 1447292. Further reading, edit. Innovative Training Network Perico. Schrader M, Costello J, Godinho LF, Eilinger M, 2015. Peroxisome Mitochondria Interplay and Disease. J. Inherit Metab Dis. 38, 4681702 do. 10.1075/S105-015-9819-7 HDL 10871/17472. PMID 256871155. S2 SID 243927113. Schrader M., Fahimi H.D., 2008. The peroxisome, still a mysterious organelle. Histosome cell biol. 129. 4421442, 10.1007/00418-008-03969. PMC 2668598. PMID 182747771. Effelsberg D., Cruz Zaragoza LD, Schlips W., Erdmann R., 2016. PEX 9P is a novel yeast peroxisomal. Import receptor 4.1 proteins. Journal of Cell Science. 129, 21 4057 4066. Do. 10.1242/JCS.1952771. PMID 276784887. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Yafraki, Quartzman SG, Dahan N, Maskit S, Zadar L, Whale U, Yofi, Olenda T, Schaldner M, Zilkva E. 2016. Characterization of proteome dynamics in OLET reveals a novel peroxisome targeting receptor. Journal of Cell Science. 129, 21 4067 4075 do. 10.1242/JCS.1952555. PMC 6275125. PMID 276635110. Matios RM, Leonum, Sandalio LM, Gomez M, Del Rio LA, Palmer JM, December 2003. Peroxismes. From pepper fruits, capsicum and UML. Purification, characterization and antioxidant activity. Journal of Plant Physiology. 160, 12 1507 16 do. 10.1,078/017/1617-01008. PMID 147174445. Corpus FJ, Barroso JB, 2014. Functional implications of peroxisomal nitric oxide, NO, in plants. Frontiers in Plant Science. 5, 97. Do. 10.3039/FPLS.2014.00097. PMC 3956114. PMID 246725535. Corpus FJ, November 2015. What is the role of hydrogen peroxide in plant peroxismes? Plant Biology. 17, 6 1099-103. Bib code, 2015 PL Bio 17.1099C. Do. 10.1111/PLB.12376. PMID 262427008. This article incorporates public domain material from Science Primer. NCBI. Archived from the original on 8 December 2009. This article incorporates text from the public domain FAM and Interpro, IPR 006708. External links, edit. Peroxisome DB, Peroxisome Database. Peroxisome KB, Peroxisome Knowledge Base. Innovative Training Network Perico. Categories: organelles metabolism. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF.